Is that what happens? So it's not.
I'm just guessing.
I don't know. I don't know. I'm not getting. Shouldn't I be getting a blink? Oh, it is blinking. Okay, that's good. All right, so let's give everyone about 5 minutes or so. Let me check who is in the waiting room. Kathleen. Hey, Kathleen. All right, so I know it's recording and most of you are here. I put the second test up last night, but apparently it was blank. So I just went in and redid it. So that should be up under the test folder. I checked it under edit and under student mode, and it is up there. So if there's a problem, I think, was it Kathleen or Jude that tell me today? Somebody or Michelle?
No, it wasn't me. I think it was Michelle.
Michelle, yeah, so I got that message, so I did fix it. Okay, so that was, you know, a waste of 2 hours of my time last night. But,
Oh, man.
Okay, so.
Thanks, Dr. Phil. We appreciate it.
Like, it's crazy that I pushed the wrong button. What? All right, so I guess I'll get started because I'm recording this. Did anybody check the videos today by chance?
No, I haven't had time.
No, it's fine. I'll check them under student mode again, but I redid them in, I want to say I condensed them into MP4 format and re-uploaded them to the bottom of lessons. It should say like Zoom meeting or something. I was playing around with those last night. So that should work, all right? They're on my computer. I just don't know why they won't condense and upload. It doesn't make any sense to me, but what do I know? So I want to go over strep. Now, yesterday we went over staph, all right? So remember, staph has all those virulence factors. And does anybody know what's diagnostic for staph?
Gram-negative?
It is not.
Is your screen?
No.
Your screen's all messed up.
Yeah,
What's
it's
It's just black.
Okay.
Yeah, there's like 2 panels of black.
That's awesome. That's perfect. All right. I will fix it. So staph is gram-positive cocci, and it's coagulase positive.
Yeah.
So new share. Still nothing?
Nothing.
So weird. Your screen share. Hold on, let me hit shop stuff. Share screen. Different, same.
Same.
You guys can see my face though.
Nope.
It's awesome.
If we have our video going, then we can see your picture. Yeah.
There's a picture that shows up, but it's not live action. It's just a
I
picture.
see
Me
that.
at school, probably.
Yeah.
Disable time. Did anything change at all? No.
Yeah, now I can
Picture,
just
yep.
do bigger. Yeah,
Now we can see your video.
that's not the PowerPoint.
Oh God.
You want me to go out there and help?
Get me a drink. That would help.
Yeah.
I'll be back in a minute, Dr. Phil.
All right.
Just
now
going to
I'm
get you that
going
drink.
anywhere.
Yeah.
Clearly. So security participants chat. Share screen. Now nothing.
Nope, still nothing.
Oh, yeah.
Gotta be one of these. I know what I want to do. How about that?
Now I see it.
Yep.
But now you
Yep.
can still see me.
Yep. I see
Yep,
you
you're
too.
on the side.
Aside.
All right, so let's try this again. All right, so yesterday we talked about staph. Today we're going to talk about strep. So these are both going to be gram-positive. Strep always means straight line. So these are going to be linear. When you look at pictures of these, if they're not completely linear, they might be in diploid, cocci, which are round, or they might be 3 or 4 in a row. So please realize when you're looking at these on, if you're doing a slide or in a picture, whatever, it depends when that slide was made, if that long linear chain was broken, or if you have some that are diplococci in 2, that just means you caught them literally in the act of binary fission, all right? So they'll show up as 2, all right? Okay, so general characteristics of strep as opposed to staph. They're gram-positive. These are spherical or ovoid, cocci arranged in long chains, commonly in pairs, because once again, they're going to be in pairs because we're literally catching them during binary fission. And remember, these are going to go through binary fission every 20 or 30 minutes, right? Just like most of the bacteria out there. Right? These are non-spore forming and non-motile, just like the staph. So these are going to be easier to kill than a spore former, and these do not have flagella, so they're not going to move. But these also have some of those virulence factors that the staph have. Now, these can form capsules and slime layers. And once again, the minute I say that, you should be thinking these are going to be slightly more virulent, because if they go through into phagocytosis by our white blood cells, it's going to be a little bit harder to get to the actual bacterium. And if they have slime layers, they tend to stick together and they tend to be able to form biofilms, which we'll be talking about, layers upon layers. So these are also facultative anaerobes. So they can live on different conditions than other bacteria. They're a little bit more virulent. They're a little bit more adaptative. All right, these do not form catalase. These are not catalase positive, all right? But they have a peroxidase system. Now, the minute I say peroxidase, you remember, these can handle oxygen, right? In oxidation or an oxygen burst. So they have a way of dealing with oxygen. Most parasitic forms are fastidious and require enrichment medium. So these don't generally grow on most things. So at school, I have them growing on blood and chocolate agar, and they sort of like a little bit less oxygen than normal. So I usually grow them in a chamber. I street plate them out, and then I put them in a chamber, cover it up, and then I light a little tea candle to get rid of most of the oxygen, and they tend to do a little better. These are small, non-pigmented colonies. So when you're looking at the colonial morphologies when they're growing, they're going to have basically no or very little color. These are sensitive to drying, heat, and disinfectants, so they kill relatively easily. Right? So they're not like a spore former. They're slightly easier to kill on surfaces, and they're really, like it says here, they're fastidious. They don't really love to grow everywhere. I mean, they'll grow, but they're not as common as like Pseudomonas or E. coli or some of the other more common bacteria that are less fastidious. All right, so that is freshly isolated Streptococcus. And usually every semester I'll have the students bring plates to work or wherever, swab bottom of their purse or shoes or phones or whatever. But invariably I have students who, one year I had somebody who worked, was an EMT, and he swabbed the inside of the ambulance, and he had the most perfect thing of strep I've ever seen. And I've had other students swab the nursing station, the keyboard, places in hospitals or healthcare, and have seen beautiful, beautiful strep. And the cool thing is, the strep, if it's new, if it's young, it stains really, really beautifully. So that's what it would look like. All right, so here's some of the questions that they'll give you in your future on different exams about specifically about strep. All right, so generally they'll ask you, how is it classified? So Rebecca Lansfield classified it according to the antigen. We've seen this word before, right? Antigen on the wall of the strep. So there's A, B, C, and so on. Another classification system is based on hemolysis. So hemolysis. So how much, when we put this on blood agar a little bit of blood, that strep, is it a beta hemolytic strep or alpha hemolytic strep? How much of that blood is it going to consume? It'll be diagnostic for the strep. I'm pretty sure if you look in the videos I put in lab, I think I did a throat swab on myself, and one of those has alpha hemolytic strep on my throat. It should be in there. If it's not, I'll find it. All right, so B for beta hemolysis, A, B, C, G, and some of the strains are D. All right, and alpha hemolysis would be like strep pneumoniae and strep viridans. And strep viridans is very, very common in your mouth, those biofilms. And generally one of the bacteria that is on the biofilms, and it usually causes some cavities and some tooth decay. So it's going to start laying its foundation on your teeth, and some of these bacteria are going to start fermenting the sugar on your teeth, and they'll produce acids and start eating away at that enamel, and they can initially start some of the cavities that go on. So if you look here, there's Streptococcus pneumoniae displaying alpha hemolysis. Right? So if you look, that auger you started out this color. I don't know if you can see the arrow, right? But as it's growing, it's consuming the blood. All right, so you'll see it disappearing. It's actually lysing the blood, hemolysis. And there's beta. All right, so the difference between alpha around the colony and beta, difference in how it would look. Right. So we're really going to be looking at pyogenes, right, that usually causes fever and a rash. Activasis we saw yesterday, that's in the human vagina. Cow udder, and I said before that a lot of these strains, we're going to be inoculating, or they are inoculating cesarean births with that. So we have the microbiome from the mother on the infant. Enterococcus faecalis, so entero usually means intestine, right? Always. Cocci is the shape, faecalis, all right, so you're gonna automatically think, well, this is in your digestive tract and in your feces. Enterococcus faecalis, human and animal intestine. Right? Strep mutans, major cause of dental caries, right here it says. So strep viridans, strep mutans, these are major causes of cavities and dental caries. Pyorrhea, some of the gum diseases that can be indicative of that too, and strep pneumoniae. Right. So it says the human respiratory tract, all right, and bacterial pneumonia, all right, not influenza. This is bacterial pneumonia. Please realize that people have pneumonia or something. Generally, this is part of your microbiome. When you really get bacterial pneumonia, usually you have something going on. So you have some upper respiratory inflammation or something's going on. Your immune system may be slightly not compromised, but it's busy. You have upper respiratory thing going on in the lungs. These are normal microbiome for you. Somehow it gets, you swallow it or you inhale it into your lungs. And then sometimes it can start bacterial pneumonia. But realize these are really, some of these are normal flora for humans in general. So like I said, we're gonna be talking about pyogenes, activasis, viridans, strep pneumoniae, and Enterococcus faecalis. So these are the ones back here. Just the ones that are bold are basically. So there's other ones, but these would be the ones you'll see, or the ones they'll ask you questions about on board exams or whatever in your future. Okay. So beta-hemolytic strep. So maybe, we've all heard of, somebody has a sore throat, and we say, well, is it beta-hemolytic strep? It's the most serious streptococcal pathogen. Right? It's a strict parasite. All right, so that means this thing literally can't really live on surfaces. It has to go from host to host. It really sort of needs a human cell with blood to live. Right? It inhabits the throat, nasopharynx, and occasionally the skin, but not for very long generally. Okay, it's a strict parasite. And it can't even make its own food generally. It has to have something else to live on or consume. Okay. So here are some virulence factors. All right, so virulence factors, when we talk about these, and I'll bring it up over and over again, these are specific things that are unique to this or this genus or species of a microbe that makes it different and makes it have some kind of advantage over other strep or other bacteria. All right, so these produce surface antigens. All right, and we talked about this with the immune system. So these are specific antigens to this. So let's just say that your immune system was able to see one of these, one of your antigen presenting cells was able to grab one of these antigens and present it to your CD4 cell and your B cells, and they were able to make antibodies to it. Your T cells were programmed for it. they'll be reacting to any of these markers, these antigens on here, right? So it has C carbohydrates, protects against lysosomes, all right? So there's this great big carbohydrate, this like C carbohydrate here is kind of like a candy coating. So even if it was consumed by a macrophage and that macrophage is excreting all kinds of lysosomes or enzymes, it has this very hard protective shell. I mean, we may get to it eventually, but it's going to be something that these lysosomes have to get through before they actually get to the cell membrane or the peptidoglycan layer here. So it's like a regular gram-positive thin cell membrane that has that thicker peptidoglycan layer. But on top of that, it has carbohydrates. All right, the other thing it has, I'm going to skip ahead. Right here, it has this prong, this M protein here, all right? So you want to picture this all the way around this entire bacterium. So even if this thing is consumed, the lysosome and the macrophage has to get through all of this. It's going all around the entire circumference of its bacteria. So it's very, very protective for it. All right, it also has fimbriae. And we mentioned these in the beginning of the semester. Those are like those burrs when you're walking in the woods or whatever that stick. And fimbriae, like E. coli has it. The bacteria, they literally adhere to whatever they're growing on. It's kind of like Velcro. They're sticking right to it. Right? These also have a hyaluronic acid capsule. Right? And I know I mentioned this before. I mean, I've talked a lot this semester. Provokes no immune response, all right? So this capsule around it is made out of hyaluronic acid. Does anyone want to know where in your body that you would normally find hyaluronic acid? Okay, so maybe you guys forgot, but that's the basis for cartilage. Cartilage and your pre-bone starts out with hyaluronic acid. So when your immune system comes across something that has hyaluronic acid on it, it doesn't think anything of it, right? It thinks it's normal because it thinks it's part of its own body. All right, C5a protease, maybe you guys have heard of protease inhibitors. It's an enzyme. Hinders complement and neutrophil response, right? So this literally inhibits a lot of the complement. And I said you guys only have to really learn about opsonization and neutralization, that complement system. So it literally hinders those from attaching. It hinders the neutrophils and everything from supermarking or super tagging this bacteria, right? So once again, it has a lot of virulence factors that help it stay alive or kind of help it dodge the immune system per se, right? Some other virulence factors of this. Now, these are things that were on bacterium itself, okay? These are actually toxins, and we talked a while back about exotoxins and toxic shock syndrome and some of these things, and I said these are gram-positive bacteria generally have exotoxins, so they're able to secrete these toxins while they're alive. Before, we talked about gram-negative, and I talked about LPS, lipopolysaccharide, and how that causes severe damage when the gram-negative bacteria dies, it secretes that. And we'll talk about that after the gram-positive. We'll talk about the gram-negative, come up again. But these are gram-positive. These are extracellular toxins. They're exotoxins. They're through exocytosis. They're released in the environment. All right, so strep produces streptolysin. So, think about what that means. Strep lysin, it's going to cut or cleave something. So those hemolysins, streptolysins, right, cause both cell and tissue injury. So this is going to release an enzyme that's going to start cleaving or cutting the red blood cells. And we said before, we talked about a little bit about alpha hemolysis or beta hemolysis. It's going to help cleave or cut those red blood cells so the bacteria can consume whatever in that red blood cell it finds or deems necessary for growth. Erythrogenic toxin. All right, erythro. Whenever you see erythro, you should automatically think red. All right, so if you're thinking red, you know, I want you to think, well, red, what could that possibly mean? Well, it's going to mean inflammation, or it could be increase in temperature, right? In this case, it means both. Erythrogenic toxin, pyro, like a pyrex, heat, pyrogenic. So it's going to induce a fever and a typical red rash. So generally, they're always going to give you a question in the future about strep throat or B-hemolytic strep or strep pyogenes. All right, so it's usually always is not quite diagnostic, but that patient's going to come in with a red throat. They're going to have a fever, and they're typically going to have a rash. You can say, well, I'm not 100% sure, but that sure as hell looks like strep pyogenes to me. And then superantigens, strong monocyte and lymphocyte stimulants cause the release of tissue necrotic factor. And I know when I was talking about the immune response, and I kind of alluded to it when we were talking about the type 4 response with T cells, some of these bacteria are going to release these toxins, and they're going to really incite your T cells to go absolutely crazy. So they're going to start attacking things they normally would. Remember, T cells are programmed to fight that very specific antigen on the surface that it was presented to. We had a formal introduction from the CD4 helper cell. Well, with these super antigens in the area, it makes the T cells go crazy. They don't quite think straight, and they start attacking pretty much everything in the area. Some other extracellular enzymes, streptokinase, all right, digest fibrin clots. And I asked you guys yesterday, all right, you know, where would this be advantageous? And Robert said, well, digesting blood clots, absolutely. So we're gonna, if we can market this or grab it, we can give it to people to digest, I can't think of the name of the medication, but let's say somebody's having a heart attack, or they're having a stroke, or they're having some kind of ischemia. We can give them this, start digesting any kind of blood clots in the body. They may not have been caused by a bacterium, but the effect is still the same with this. Hyaluronidase. So hyaluronidase, it's an enzyme. And I said hyaluronic, uh-oh. Did you guys just see that? Is everyone still there?
Yeah.
Yep.
Yep.
Here, I'm like,
It's gonna shut us down in 9 minutes.
I literally, I put 2 calls, and I'm like, I want to get this. I just want to know what I have to do. He never got back to me, and then in the middle of my whatever. Oh my God. All right. So anyway, so hyaluronidase breaks down connective tissue. So we said before that this bacteria has hyaluronic acid all around it, so the immune system doesn't even know it's there, and it can release an enzyme that can break down connective tissue. So this is another one of those bacterium that can release this enzyme, and it can literally digest its way through anything it's near. So let's say there's around connective tissue, cartilage, the peritoneum or whatever. It just releases an enzyme. It'll just start eating its way through whatever it comes in contact with, right? Sounds terrible, but think of how cool that is if you're a bacteria, right? DNAs we saw yesterday, hydrolysis DNA. So these are common with the staph or the strep. Very common virulence factors. All right. So humans are the only reservoir for this. All right. So you can make out with your dog or cat all you want. They're really not going to have strep pyogenes in their throat. And some people are inapparent carriers, and we talked about this in the beginning of the course. So, like if I have a sore throat, sometimes I have it. I am asymptomatic. I don't know I have it, but I have done in the past probably 5 years, so that would be 15 or 18 classes, I've had it 4 times. Didn't even know I had it. Transmission is through contact, droplets, food, or fomites. Port of entry is generally the skin or the throat, pharynx. And children, we all, if you have children or nieces or nephews or whatever, children are a predominant group affected for cutaneous and throat infections. Kids have a strep throat, right? Systemic infections and progressive sequela, possibly if untreated. So this is another one that we want to treat because our body is going to make antibodies. And I said when we saw that capsule, I said your body's going to make antibodies to it. And sometimes these antibodies will look just like other parts of your body, and your antibodies that you made to this will start attacking your own tissues, and that's not a good thing. So skin infections from staph also can cause a lot of these other cutaneous infections. So usually when we think of staph, we think of the throat. Generally, this is going to be something on the skin. So vertigo is superficial lesions that break and form highly contagious crust. Often occurs in epidemics in school children. And then with an insect bite, you've got an open cut, staph gets in there. Poor hygiene, people aren't showering or living in crowded living conditions. And this thing just looks like a potato chip to me. It's just disgusting. They literally peel right off. They're crunchy. Gross. Virulence or pathogen enters through a break in the skin. All right. Eventually spreads to the dermis and subcutaneous tissues. Can remain superficial or become systemic. All right. So this is actually the strep. If you look in here, it's releasing those toxins. It's starting to erupt in the skin. Right. You can see that with the red here. And here's a streptococcal inflammation of the pharynx. This is strep throat. Right? So if you've ever seen this in kids, kids have these huge tonsils. They're, screaming, they're grabbing their ears or throat, or whatever you look in. It's completely red and inflamed. But generally with this, once again, they're going to have fever, and they're generally going to have a rash. Not always, but most times. All right, systemic infections are called scarlet fever. Obviously it gets its name from scarlet color of that, right? Strain of Streptococcus carrying prophage that codes for erythrogenic toxin can lead to sequela. All right, so this is it's a strain of this, and the child might have something else going on, or might have a weakened immune system, or something else is going on. The strain can take over. Maybe there's not a lot of other bacteria in the microbiome, so there's no competition. Can lead to septicemia, which we said before was reproduction in the bloodstream. Pneumonia, all right? I said before, people just generally don't come out of the blue. You don't wake up with pneumonia unless you got something else going on in most cases. So if they had allergies or rhinitis or just say a common cold or something, and the immune system was being occupied, or they had a runny nose, or their throat was dripping or whatever, then some of these strep pneumoniae can get into the lungs and start causing it. You can cause streptococcal toxic shock syndrome. So it's toxic shock syndrome caused by this specific genus and species. All right. And this is what I talked about the other day, and I said, this is a sequela. We learned that word day 1. So sequela is something that happens, you have the primary condition, whatever it is. This might be scarlet fever or sore throat or whatever it happens to be. All right, so you have that. This is an outcome that can happen just because you have the initial condition. Rheumatic fever Follows overt or subclinical pharyngitis in children, carditis with extensive valve damage possible, arthritis, chorea, or fever. So I said before, your body's going to make antibodies to that strep. All right. Unfortunately, the strep antigens look very similar to the HLA markers or the MHC markers on the endocardium. So it starts really attacking itself here. Not good. So you have vegetations or things growing on this. Right? So usually, if they give you on your board exams or NCLEX, if they give you a case study, like a child, 8-year-old child, comes in with sore throat or whatever, and then 6 or 8 weeks later, they'll give you a time frame within 90 days or whatever. Child complains of, they'll give you some example of a heart condition, like the child was lethargic or out of energy or something. You'd have to think, well, what could cause it? Respiratory, okay, heart, cause heart. Well, sore throat, 90 days later, they had something to do with hypoxia or diminished blood flow or something. Okay, well, I'd have to rule in or rule out rheumatic fever at some point for that, because it's a sequela, right? Acute glomerulonephritis. All right, so acute means sudden. Glomerulus, your nephrons are the the functioning portion of your kidneys. All right, itis is inflammation. So acute inflammation of the glomerulus and the nephrons doesn't sound good at all. Nephritis, increased blood pressure, occasionally heart failure, can become chronic, leading to kidney failure. All right, so you guys remember your juxtaglomerular apparatus? All right, if you don't, I suggest you write that down. All right, the juxtaglomerular apparatus, all right, the macular densa cells, those are what regulates all your blood pressure. So when anything jams up your glomerulus or your kidneys are not getting ample blood supply, all they're going to do is secrete, remember that renin, angiotensin 1, angiotensin 2 cascade? All right, something you should be very familiar with, because once again, your kidneys are going to regulate the majority of your blood pressure. You get this thing inflamed or irritated, kidneys, you're not making enough filtrate. Our kidneys don't know what's going on. They're saying, I'm not making enough filtrate. There's not enough blood going to the glomerulus.